let's chat about something that everybody probably has in mind all the time, vaccines. So I'm not going to do the regular 40-minute rant about vaccination in general. So in regards to vaccines, what you need to appreciate the different types of vaccines, and I provide the list. And of course, this list is now incomplete. But don't worry, I'm going to ask only about the vaccines that are on the list in the notes. I'm just going to give you an idea what each type of vaccines entails. So the most probably common and well-known are inactivated vaccines, in which, well, pathogen is inactivated. We don't say killed because viruses are not living, so you can't kill something that is not living. An example of an activated vaccine can be an injectable polio vaccine, for instance. I can probably come up with something else. Smallpox is different. Smallpox is attenuated, and that's gonna be actually next on the list. There's a bunch of like experimental inactivated vaccines. For instance, there is an inactivated vaccine against COVID, which is produced in Russia by the company that I used to work for, actually. Attenuated vaccine. So in this case, pathogen is, well, weakened. So here comes smallpox, Julian mentioned, absolutely. Yellow fever, oral polio vaccine is attenuated, the Sabin vaccine. Measles, mumps, and rubella are attenuated vaccine. All of them are attenuated. You don't need to know the example, just understand like what the word means. And then we have subunit vaccines. So basically in this case you take an antigen is subunit or recombinant, sometimes they're called. So you have an antigen, it's a protein in basically all cases, is expressed independently. So classic subunit vaccines include human papillomavirus and hepatitis B vaccine. These vaccines are usually highly effective, extremely safe, and very, very pricey. Among the COVID vaccines, subunit vaccine is Novavax, which is awaiting for the emergency use authorization from FDA. Then we have vector vaccines. So in this case, a gene for an antigen, and when I say antigen, we're talking about a target antigen, the one that we want the immune system to respond to. So this gene is inserted into, I put it in the brackets, usually viral vector, okay? So the first vaccine that was actually used publicly was Ebola virus vaccine based on the adenovirus vector. Now Johnson & Johnson, or Janssen vaccine, it's better to say Janssen because that's actually the vaccine branch of the company. AstraZeneca vaccine and Sputnik V, Russian vaccine, they all are based on the different adenovirus vectors that contain the gene for the spike protein. So basically, the viral vector, the adenovirus, is completely gutted. It can't replicate, it cannot hurt person. Does that make sense? But it can still deliver the gene, and gene will be expressed. Then we have DNA or plasmid vaccine. A gene for antigen is in the plasmid. And now I want to make it very clear. So these 6 are Why there are 5? Probably forget something, but no, it seems genuine. No, that's 5. So there is another type that appeared this year, and it is a seismic shift in vaccinology. Mrna vaccine. So a gene is an mRNA molecule packaged in a lipid nanoparticle. Okay, now, a few comments. Inactivated vaccines are usually extremely safe, but in most cases they lack T-cell response, good T-cell response at least. They produce good antibody response, and often you can get away with it, but T-cell response is usually poor. Attenuated vaccines, on the other hand, usually give a good T-cell response. But the problem with attenuated vaccine is that, well, it's a weakened pathogen. So for smallpox, for instance, there were cases recorded when there was actually a death associated with it, when a person who received the vaccine as he was in the military, turns out he was immunosuppressed and he died. Because it is weakened for a healthy person, but if you are not healthy, it can blow out of proportion. Subunit vaccines already mentioned, probably the only downside is expensive. Vector, now we have several vector vaccines against COVID. They're not approved. I'll talk about approval, just mention it quickly. DNA vaccines. The main concern for DNA vaccines is that DNA, as a plasmid, will incorporate in the chromosomes of human cells. Does that make sense? Now, I say will because we know that it will. It was shown that it will incorporate it in the chromosome. And the main risk associated with that, of course, is disruption of certain genetic signatures, for instance, disruption of tumor suppressor genes or stimulation of oncogenes and subsequent transformation of the cell. Every time you mess with the DNA, you're running the risk of, getting to cancer. So this is why people are kind of hesitant. Does that make sense? Somebody would tell me a year and a half ago, well, 2 years ago, okay, that there will be a vaccine based on mRNA, I would laugh in their face. From biochemical standpoint, that wouldn't be possible. The first mRNA vaccine, the Mrna vaccine, was developed from receiving the sequence of the virus to first prep that went into trials on animals, like first actual prep. 63 days, which is mind-boggling. Absolutely I could not imagine anything like that. Getting a vaccine from the sequence of the fucking gene into humans, and I'm not talking trials, I'm talking distribution. The sequence was in January, the first doses were administered in December. This is crazy. 10 months. It's never been done. And now there are mRNA vaccines, and they induce not only antibodies, as we all know, but they also induce a very robust T-cell responses, which means they not only can prevent the infection, but it also can clear the infection. Does that make sense afterwards? And now there are vaccines against CMV in the pipeline, respiratory syncytial virus, influenza. So possibly we won't get influenza vaccine every year finally. And just a couple of months ago, the paper was published with a fairly successful vaccine against malaria. Experimental, of course, but I think they already start rolling it out in a clinical trials. Now, a little sort of public service announcement, it's not for the exam, but you hear vaccines get a lot of undeserved flack, and one of the arguments is that, these coronavirus vaccines never received an approval. What is the difference between emergency use authorization and approval? Basically, the only one component of data that is lacking in emergency use authorization is a long-term efficacy. Long-term. We're talking about years and years. And why? Because it was just a year and a half. It's impossible to get those data yet. Does that make sense? Safety, immunogenicity, all of this was absolutely storage. Everything is fine. We just don't know how long the immunity lasts to finally approve it. Does that make sense? If at the end of the day, like 2-3 years down the road, Pfizer, Mrna, Janssen, they all show that yes, immunity lasts for 3 years, they might apply for approval and we're going to have approved vaccines, fully approved, just like MMR. Does that make sense? Any questions? Okay, that's sad. That's my favorite topic.